Hello, everyone. I'm your host, Jeff Henno. I'm an Associate Director for Precision Health Marketing at PacBio. I want to welcome everyone to the PacBio webinar series, with today's topic focused on tackling challenging genes and their paralogs involved in recessive disorders. I'm joined today by my colleague, Dr. Zhao Chen, a principal scientist in our computational biology group. We're both very excited to share a lot of great material with you today. Please note that our presentations have been pre-recorded, but will be followed by a live Q&A. You are welcome to submit your questions at any point during the webinar by typing them in the provided area on your attendee control panel, and we'll get to the questions at the end. We've also uploaded a number of complementary pieces of literature to your control panel, so feel free to download those. We're recording this entire webinar and we'll be making it available for download in the next few days. Please keep an eye out for a follow-up email about that recording. So with that, let's get started. I will begin with a general overview of PacBio solutions for the analysis of medically relevant and challenging genes, and then we'll dive into the bioinformatics portion with Dr. Chen. I have 3 topics during my talk. We will have a brief look at our 2 HiFi systems. Then we'll ask ourselves a question: why are some genes challenging to resolve? With those challenges in mind, we will then address some new sequencing-based solutions that will be helpful in the field of recessive disorders and beyond. First of all, we're quite proud of the fact that Nature Methods has selected long-read sequencing as method of the year in 2022. One of the main arguments was a telomere-to-telomere paper, published last year and addressing the last 8% of the human reference genome that remained very challenging to map. Interestingly, Dr. Karen Miga, one of the key authors of the paper, stated that the feat probably wouldn't have been possible without new sequencing technology from PacBio. Let's have a look at our 2 HiFi sequencing systems. On the right-hand side, we have our well-established Sequel IIe, launched in 2019. On the left, you can see our latest system called Revio, which we announced last year and will start to ship in the coming months. This system has 4 independent stages, each running a smart cell with potentially different applications. Revio has a much higher throughput and has already seen a lot of interest from the field. But today, we'll not focus on the sequencing part itself, but rather look at solutions pre- and post-sequencing in the light of these challenging single-gene disorders. So why are some genes challenging to resolve? The sequencing itself can pose challenges due to high GC content, microsatellites, short tandem repeats, etc. Also, there are mapping challenges related to paralogs, pseudogenes, segmental duplications, and copy number variation. Some genes contain very complex variants, such as SVs, CNVs, gene fusions, and gene conversions, to name a few. Phasing is often needed to better understand the disease genetics. Without this information, one must often rely on pedigree analysis. And lastly, the methylation signal is key for many disorders. Here are some typical examples of genes and known NGS challenges which can be resolved by implementing HiFi sequencing. We will have a closer look at some of those genes today. So how do we circumvent those challenges? We like to take a comprehensive approach with targeted solutions. Obviously, a lot of these aspects we'll discuss today also apply to genome-wide approaches, such as whole genome sequencing. Some challenges can already be addressed in the enrichment phase by capturing or amplifying large inserts for HiFi sequencing. This allows to obtain gene-spanning reads and to look at hybrid alleles, for example. Capture probes can be tiled across the region of interest. We'll discuss this later. In a second step, HiFi sequencing with long insert sizes up to 15 kb provides high accuracy, which significantly improves the read mapping quality, and gets you the robust coverage needed for variant calling. In recent years, PacBio and third-party players developed a suite of advanced analysis tools, which allow the researcher to select the right tool set for the job and uncover more genetic context than ever before. You can now look at all variant classes, including small to large structural variants. You also get the ability to phase the variants, analyze and visualize short tandem repeats, understand copy number changes in areas of segmental duplications, identify pseudogenes, and so on. Let's look into method consolidation for those typical genes involved in carrier screening. Targeted HiFi sequencing workflows are increasingly implemented to consolidate legacy molecular methods. Here's how this concept looks. In many screening and research labs, variant detection is carried out by combining several molecular methods per gene or disorder, as you know. This can be time-consuming, complex, and costly. Each method detects a subset of variants, often focusing on known variants only. We propose to analyze these genes by streamlining the data generation into one robust long-read solution to detect all key variants. The sequencing is done via targeted approaches, which are robust, scalable, and cost-effective. This allows the researcher to select tailored bioinformatics tools in the backend. We are convinced this concept is a more robust approach, providing a lot more insights and utility without increasing the cost. By the way, our informatics tools are all freely available. Here are a number of PacBio customers who are actively implementing this concept. In the next slides, I will discuss solutions for target enrichment, followed by tools for the data analysis. Let's look at amplicons. As shown here, typical amplicon sequencing workflows that use short-read NGS or Sanger sequencing require complex assay designs. With HiFi sequencing, you get a simplified workflow with a single primer pair and reads that span the entire amplicon. As such, HiFi amplicon sequencing is recommended for single genes or smaller panels, and PacBio has the barcoding and library preps to support you. Here's an example of how simple the design can become for GBA. Two primer pairs are used to amplify GBA and its pseudogene with amplicons of 12 and 15 kb, spanning both genes easily. We have an app note for you that describes this amplicon approach for GBA. We'll talk more on that later. To offer a robust solution for larger gene panels, say above 10 genes, we have partnered with Twist Bioscience to enable enrichment via hybrid capture. Twist probes are very high capture efficient and allow exceptional uniformity. Twist and PacBio work together to develop long-read workflows with optimized probe tiling, requiring fewer oligos and offering strong performance in GC-rich regions. On the right, you see how Twist offers a custom panel design, which is free of charge, custom probes, and optimized library prep reagents, compatible with downstream sequencing and analysis from PacBio. Now that we've looked at the enrichment side, let's jump into the bioinformatics for a second. Here's an overview of a new snake-make solution bundling our existing tools to go from demultiplexing to annotated variant calls. This setup is easily configurable with any number of amplicon targets and can be extended with additional specialized tools like PacBio or Paraphase. We'll discuss this later. In the remaining slides, I will show some examples from the research field where HiFi reads were implemented in a targeted approach for congenital adrenal hyperplasia and for thalassemia. Let's start with CAH, where CYP21A2 is a central gene. The paper on top discusses how all previously detected pathogenic variants were detected in 48 control samples by utilizing an amplicon approach with HiFi. The bottom paper was actually published by our customer Berry Genomics and discusses the performance of their comprehensive HiFi sequencing approach for CAH, also with long-range PCR as enrichment. In the field of thalassemia, similar approaches have been published. Here's a selection of 2 recent papers, but there are many more. The top paper discusses the performance of targeted sequencing for HbA1 and 2 and HBB to allow for a universal analysis approach. The second paper by Liu et al. Looks at detection rates of their amplicon solution versus conventional methods and observes almost a 10% increase in positive detection rate of pathogenic variants across over 430 cases. We recently published several app notes, and more are being developed. These documents describe the main aspects of our targeted sequencing solutions for key disease areas like thalassemia, congenital adrenal hyperplasia, and Gaucher disease. The app notes contain more details about the PCR setup, the library prep solutions, and downstream analysis options. Feel free to download them from your attendee control panel. Okay, so in a nutshell, here's what we've discussed. First of all, HiFi sequencing allows to obtain robust coverage of regions that are difficult to sequence or difficult to map. You also get haplotype resolution through direct phasing. Our accurate reads allow to call variants across all classes and the discovery of novel and rare variants. For targeted sequencing, our motto is, if you can amplify or capture it, PacBio systems can sequence the region full length. Also, these workflows are cost-effective and very scalable. To give you an idea, typical costs for the PCR, library prep, and sequencing will range around $50 per sample, depending on the sequencing system and the multiplexing level. This cost excludes the DNA extraction. A higher multiplexing allows for lower cost per sample, down to around $20 per sample. These are indicative prices. Lastly, our concept of workflow consolidation allows for a more streamlined and less complex approach per gene or disorder. Thank you very much. I will now hand it over to my colleague, Dr. Xiao Chen.
Hi, everyone. My name is Xiao Chen. I'm a bioinformatics scientist at PacBio. Today, I'll be talking about how we resolve highly homologous genes with PacBio HiFi sequencing. As Jeff mentioned in his talk, a lot of the medically challenging regions have to do with high sequence homology and complex structural variants. PacBio amplicon sequencing provides a straightforward approach to analyzing those challenging regions in single amplicons. But for shotgun sequencing, whether it's WGS or target enrichment data, we still face informatics challenges in those regions. Today, I'll be talking about how we solve such problems with a new informatics tool. The problem that we are dealing with here is the so-called segmental duplications. These are long sequences that are present more than once in the reference genome. Segmental duplications, they comprise 7% of the human genome. If we look at the figure on the right here, this shows the intrachromosomal segmental duplications in our genome. The black ones are those ones already known for GRCH38, and the red ones are those that are newly found in the new CHM13 genome. Many of the clinically relevant genes fall into these segmental duplications. These are highly dynamic regions where so-called unequal crossing over often happens, creating lots of large structural variations, including deletions, duplications, and gene conversions. It's very important that we should be able to detect these large events together with small variants. But in these segmental duplication regions, the high sequence similarity creates lots of problems to read alignment and variant calling. The HiFi reads are long and highly accurate, so they are ideal for solving these homology problems. But we still need new informatics tools to solve those regions where the homology is longer than the HiFi read length. So we developed this tool called Paraphase. The idea behind it is that when we talk about genes that have highly similar gene family members, the sequence similarity is so high so that we can analyze them as if they are different copies of the same gene. For example, here we're looking at the green gene of interest, and it has a highly similar paralog and a highly similar pseudogene. The variant, if we look at the read alignments here, because of the high sequence similarity, the alignments are ambiguous and sometimes wrong. What Paraphase does is that it extracts reads that align to all the relevant regions here, and then realigns all of those reads to our gene of interest. After realignment, it will phase those reads into haplotypes, and each of those phased haplotypes will be analyzed for variants. The red star symbol here is an example of a pathogenic variant on the gene, and the green oval is another example of a gene conversion on the paralog, which is a region where the sequences are identical to the corresponding region of the gene. And copy number variants are also possible in these regions. For example, for the pseudogene, there's only one haplotype found in this example here, suggesting that there is a deletion. So after each of these haplotypes are annotated for variants, we can infer the copy number of the functional gene and the disease and carrier status of a sample. So I'll be talking about how we apply Paraphase to some of the medically relevant challenging genes. And I will start with an important gene called SMN1, and then we'll talk about applications in other genes. Mutations in SMN1 cause this disease called spinal muscular atrophy, shortened as SMA. It is a leading cause of infant death, a very important disease. And SMN1, in the reference genome, it has a highly similar paralog called SMN2. The two genes are nearly identical in sequence, except one functionally different base at the so-called C.840 site, where SMN1 has a C base and SMN2 has a T base. The T base in SMN2 promotes skipping of this very important exon 7, resulting in almost non-functional transcripts and very low protein levels. The majority of SMA is caused by biallelic loss of the C.840C, either through large deletion or gene conversion to the T base. And a small percentage of SMA is caused by other pathogenic variants in SMN1. Conventionally, SMN1 testing is done by testing the copy number with PCR-based methods, which are mainly targeting the C.840C site. And the copy number of SMN2 is also medically relevant, as it's a modifier of the disease severity. In recent years, high-throughput sequencing-based methods, testing methods for SMN have started to emerge. But these two genes are 30 kb long with very, very high sequence homology, making it almost impossible to resolve by short reads. And even for HiFi reads, we are facing many mapping problems, as we can see later. For SMA, both newborn screening and carrier screening are recommended by American College of Medical Genetics and Genomics. If we look at this region on an allele, it could have variable copy number of SMN1. Most often 0 copy, 1 copy, or 2 copies. And these are all based on the C.A40C site. And for each of these copies, it's also possible to have a pathogenic variant at the site that's not C.A40. So we need a test that can comprehensively profile SMN1 and SMN2. And this needs to include the identification of, first, the copy number of SMN1 and SMN2 based on C.A40. This is also the focus of most conventional SMN testing assays. But beyond copy number testing, we also need to be able to identify pathogenic variants throughout both genes. And also, in the space of carrier screening, it's very important to identify the so-called silent carriers. Silent carriers are those that carry 2 copies of SMN1 on one chromosome and 0 copy on the other. So these individuals will have a total SMN1 copy number of 2. So they are easily misidentified as a normal individual. And silent carriers, they account for more than a quarter of carriers in African populations. So they are not really that uncommon. Currently, it's impossible to identify these silent carriers without pedigree information, as it's very difficult to physically phase through this very long and complex region. Currently, people are using a marker SNP that has previously been found to associate with 2-copy SMN1 alleles. This SNP is one that's common in African populations, but its accuracy is relatively low because it's present on both single-copy SMN1 alleles and 2-copy SMN1 alleles. For example, in an African individual with 2 copies of SMN1, if this individual tests positive for this marker SNP, there is a 1 to 3% chance that this individual is a carrier. We really need better markers for silent carriers. But again, because this region is so challenging to analyze, we're currently still not able to find good markers for silent carriers without good analysis tools. So we can see that for SMA testing, we need comprehensive SMN1 and SMN2 profiling, which includes calling the copy number of SMN1 and SMN2. And beyond copy number testing, we need to call the pathogenic variants and the silent carriers. For SMA research, we also need new tools to analyze this region so that one can better understand why some SMA patients have less severe symptoms than other SMA patients. And this often has to do with the disease-modifying effects of variants, especially in SMN2. In addition, this is one of the most complex regions in the human genome. And allele can often carry like multiple variable copy numbers of SMN1 and SMN2. And it's the full structure of this region is very hard to resolve as it involves megabases of sequences and multiple groups of segmental duplications. It is very important to be able to characterize the structure of this region so that we can learn more about the mechanisms leading to the complex structure and the evolutionary forces behind. So now let's take a look at this region in short-read data versus HiFi data. So here, take this sample HG01175 as an example. The top panel shows the short read data, and we can see that the majority of reads map ambiguously with low confidence. So 84% of reads map with a map Q of 0, suggesting that the aligner is not able to distinguish SMN1 from SMN2 in the majority of reads. In the second panel here is the raw alignments of HiFi WGS data. The map Q, the mapping qualities are better than short reads, with the majority of reads having a map Q higher than 0, but still just slightly over half of the reads have a mapping quality higher than 5. And sometimes SMN1 reads will misalign to SMN2, and SMN2 reads will misalign to SMN1. So in total, about 19% of reads misalign in this way. As a result of the misalignment and the low mapping quality, this region couldn't be phased into 2 alleles. Now for Paraphase, it takes reads that align to either SMN1 or SMN2, realigns the reads, all of these reads to SMN1, and then it will take all the variant sites and phase these reads into complete haplotypes. And once we have the haplotypes, they are assigned to SMN1 or SMN2 based on the base at C.A40. And here, all the reads are assigned to the haplotypes that they come from. So we can see that this sample has 2 copies of SMN1 and a truncated form of SMN2 with a deletion of the last 2 exons, and followed by an intact form of SMN2. Once we have the haplotypes, it will be very straightforward to determine the copy number of SMN1 and SMN2 and make phase variant calls. So this easily fulfills the goals of first, calling the copy number, second, identifying any pathogenic variants naturally from these phased haplotypes. We benchmarked our copy number calls against orthogonal methods, and our concordance is 99.1% for SMN1 and 100% for SMN2. Being able to completely profile the SMN1 and SMN2 sequences has enabled us to do some cool research on this region. So here we took complete haplotypes from SMN1 and SMN2 across 5 different ethnic populations and did a population-wide haplotype analysis. On this tree on the left here, we can see that the SMN1 haplotypes are separated from SMN2 haplotypes. So all the SMN1 haplotypes are to the left of this red dotted line in the middle, and all the SMN2 haplotypes are to the right. And these haplotypes, they are further separated into smaller haplogroups that share the same common ancestor. And we name these haplogroups S1-1 through S1-10 for SMN1 and S2-1 through S2-9 for SMN2. The representative sequences from these haplogroups are shown on the right. And here we can see that between SMN1 and SMN2, the sequence differences are mainly located in the last 2 exons and also the downstream regions. And SMN1 and SMN2, they are essentially indistinguishable in the first 6 exons. We use pedigrees to phase these haplotypes into alleles and quantify the allele frequency across the 5 ethnic populations. Particularly, we are interested in which haplotypes form the so-called singleton SMN1 alleles that have just one copy of SMN1, and which haplotypes form the 2-copy SMN1 alleles, which are alleles that carry 2 copies of SMN1 on a single chromosome. Through this analysis, we identified a common pair of SMN1 haplotypes, S1-8 plus S1-9D. This allele comprises 2/3 of African 2-copy SMN1 alleles. And each of these 2 haplotypes are really present as singletons. So they serve as a potential marker for silent carriers if the pedigree information is not available. We quantified that for an African individual with 2 copies of SMN1, testing positive for S1-8 and S1-9D gives a silent carrier risk of 88.5%. This is significantly higher than the currently used marker SNP. For the currently used marker SNP, testing positive for the SNP just gives a carrier risk of about 1 to 3%. So that wraps up the story of SMN1 and SMN2. In summary, we applied Paraphase to this very difficult region and show that Paraphase is able to identify the standard carrier for SMA, and it can call pathogenic variants other than C.A40 C2T, and it enables haplotype-based screening of silent carriers. Through our population-based analysis, we found markers for silent carrier testing, and more detailed results can be found in our recently published paper. Future analysis of large population data with Paraphase will allow identification of more diverse haplotypes and more genetic markers for silent carrier detection. And complete sequence analysis of SMN1 and SMN2 will allow researchers to make new discoveries for SMA research. Now, going beyond SMN1, we have been trying to extend Paraphase to more genes with highly similar paralogs or pseudogenes. A good example is this RCCX module. This is a 30 kb long tandem repeat with very frequent copy number changes. This region contains 4 clinically relevant genes. We have CYP21A2, shown as the green box. It has a pseudogene called CYP21A1P, shown as the yellow box. CYP21A2 causes a very important disease called 21-OHD-CAH. And then we have TNXB, which is a long gene that partially overlaps this RCCX module. It's got a pseudogene called TNXA in the other copy of the repeat. And then finally, we have C4A and C4B. These are paralogs of each other. They are components. They are part of the human complement component 4, and they are relevant in autoimmune diseases. We apply the same haplotype phasing approach to this RCCX module, and now we can reveal all copies of this repeat in a given individual. Here, in this example, there are 4 copies of this repeat in total, and each allele contains 2 copies. The 2 haplotypes in green form one allele, and the 2 haplotypes in purple form the other allele. Copy number variants are very common in RCCX. Here, we're showing 2 examples here. In sample 1, on the purple allele, there is a deletion that there is a deletion of one copy of this repeat, so that this purple allele now only contains one single copy of RCCX. And the breakpoint of the deletion happens right within this gene, CYP21A2, creating a fusion gene between the pseudogene and CYP21A2. And this fusion gene is non-functional. In sample 2, there is a duplication of this RCCX module, creating an additional copy on the green allele. So now on the green allele, there are 3 copies of RCCX. And this creates an additional copy of CYP21A2 here. Interestingly, the last haplotype contains a pathogenic variant in CYP21A2. And this sample is easily misidentified as a carrier when one sees this variant, if the testing method is not able to correctly identify the duplication or phase it with the pathogenic variant. So this is an example showing the importance of CNV calling and phasing in segmental duplications. We quantified the frequency of copy number variants in RCCX across different populations. Here we can see that the copy numbers is indeed very variable in the population. And in some populations, more than 40% of the individuals have a copy number loss or a copy number gain of RCCX. And interestingly, in South Asians, the frequency of copy number gains is extremely high. Now, a second example for Paraphase would be this gene called STRC. Mutations in STRC cause hereditary hearing loss and deafness. So here we're showing an example where Paraphase detects biallelic variants in STRC. In STRC, there is a 15 kb long region with high homology to the pseudogene. And this region is flanked with distinct sequences that are pretty different between the gene and the pseudogene. This homology region is where variant calling can often be wrong, especially those very big deletions or duplications when the breakpoint happen right within this homology region. So here, the proband has only one copy of STRC, and it contains a stop gain mutation. This allele is inherited from the father, and the father has another functional STRC allele. In the mother, there's only a single copy of STRC, and this copy is not inherited in the proband. So we can see that the proband has an allele that carries a pathogenic variant inherited from the father, and another allele, which is a deletion allele, which is inherited from the mother. And the last example here is this gene called PMS2. Mutations in PMS2 cause Lynch syndrome, which is a disease with increased risk of getting many types of cancer. Here we found that there is gene conversion between PMS2 and its pseudogene PMS2CL. In the screenshot, the IGV screenshot here, in sample 1, there is no gene conversion. So we can see that the homology starts from exon 11, exon 12, where the sequences are pretty different between PMS2 and the pseudogene. And then the sequences become more similar in exon 13, 14, and finally, they become indistinguishable from each other in exon 15. In sample 2, there is a PMS2 allele, the second allele, that carries a a converted region where this is a PMS2 allele, but the sequence looks like the pseudogene in exon 13 to 14. And another example would be sample 3, where the first PMS2 allele, the sequence is identical to the pseudogene in exon 12. And the reverse gene conversion can also happen. For example, in the first PMS2 the pseudogene allele in sample 3, the sequences in exon 13 to 14, they look exactly like PMS2. So they have been converted to look like PMS2. We quantify the frequency of gene conversion across populations. And here we can see that gene conversion is actually very common in exon 13 to 14, especially in Africans. For PMS2, 25% of PMS2 alleles in Africans have been converted to look like the pseudogene. And 75% of the pseudogene alleles in Africans have been converted to look like PMS2. And we think that in exon 15, there could be an even higher level of gene conversion that has been happening to keep the gene PMS2 and the pseudogene so similar in sequence so that they are indistinguishable from each other. Now, we have shown that we have applied Paraphase to solve some highly medically relevant genes such as SMN1, the RCCX module, PMS2, and STRC. Currently, we are incorporating more clinically relevant genes that are affected by high sequence homology into Paraphase. And some examples are IKBKG, NCF1, NEB, factor VIII, and CFC1. So in summary, Paraphase provides a single framework for resolving highly homologous genes. And we are currently building it towards a genome-wide generalized color for genotyping paralogs. Researchers will be able to use Paraphase to reveal new biological insights through population-wide analysis in those genomic regions that were previously inaccessible. With that, I would like to acknowledge the people who have provided data and feedback to this work, and also thank you for your time.
All right, wonderful. We hope you enjoyed our presentations, and we can go for some questions that already came through. Please feel free to continue submitting those questions as we are answering what has already come into our list here. So, Zhao, I will read them, and so everyone can follow the question. First one, when one is conducting whole genome sequencing to try to discover potential disease, say in newborn screening, one tries to avoid false positives, which potentially results in conservatism in reporting, i.e. only report well understood variants. Thus, one will potentially miss the false negatives, particularly in less well understood or reported on populations. Does one have to adopt some form of parallel exercise, say tandem mass spectrometry, or can PacBio overcome the problem?
Yeah, when we talk about segmental duplications, if one uses standard secondary analysis tools, they will tend to make lots of false positive or false negative calls in those regions because of the high sequence similarity. That is why we develop Paraphase, which is a specialized tool dealing with those difficult regions. And we've shown that Paraphase has a much higher accuracy for calling variants in those regions, both sensitive and specific that is with regard to the informatics part. When it comes to variant interpretation, that's another story. And so yeah, so for Paraphase, where we focus on the informatics and we try to make the most accurate variant calls, and that is our strategy for segmental duplications.
Thanks. Second question, were there any other haplotype pairs?
I believe this is talking about SMN1. Yeah, so we found that one pair of haplotypes that form 1/3 or 2/3 of African 2-copy SMN1 alleles. And yes, there are other pairs of haplotypes that form that comprise the rest of the 2-copy SMN1 alleles in Africans, and also there are other pairs that are found in other populations. So we're hoping that with future studies, when we include more and more samples from different populations, we'll be able to have more statistical power to really identify more markers to say that, like, when 2 haplotypes are identified in one individual, it's more likely that they appear on the same chromosome instead of one each on 2 chromosomes. So that kind of relies on a bigger sample size. So that's why we were mainly talking about the one most common haplotype pair in Africans.
Yep. All right. Next question, does copy number calling with Paraphase still work if all gene copies are exactly the same? Or does it depend on variation between the different gene copies?
Yeah, so we do consider scenarios where 2 haplotypes are identical. So that's why we use read depth to tell apart cases where it's 2 haplotypes that are identical or just one copy. We will compare the read depths covering each haplotype against the average coverage across the genome in the case of whole genome sequencing, or we'll do certain depth normalization for enrichment type data. The idea is to see whether the read depth covering a haplotype is consistent with 1 copy of the gene or 2 copies.
All right. Another great question here. Do you plan to implement on CHM13?
Yeah, so currently Paraphase only works on GRCH38. We do have plans to adding support for both GRCH37 and CHM13, but that would be sometime in the future.
All right. So stay tuned for that, I would say. Next one, for SMN1 and SMN2, you use the variant C.840 to differentiate between the 2 genes. What about the RCCX module? There is also a unique variant. Is there also a unique variant to differentiate?
Yeah, so there are, like, for take the gene SUB21A2 in the RCCX module as an example, there are base differences between the gene SUB21A2 and its pseudogene. So we can use those variants to differentiate SUB21A2 from the pseudogene once we have the full haplotypes. And also, one idea that we have been thinking about for this type of analysis is also that when we look at these problems with genes and paralogs and pseudogenes, in some sense, one might not be really interested in differentiating the gene from the pseudogene, because, like, as we have full-length haplotypes, after variant annotation, we'll be able to easily tell which haplotype encodes the functional gene and which haplotypes encodes non-functional genes. Could be pseudogene, or in the case of SMN2, that one base with the functional, like that decreases the function of the gene. So it's more about annotation, functional annotation, rather than the physical location of these genes. So after variant annotation, we'll be able to easily calculate the copy number of the functional gene in each individual.
All right. And questions keep rolling in. We'll go to the next one. If a mixture of 10 individuals was sequenced as one sample, is it possible to phase out the haplotypes for each individual?
So Paraphase is currently designed for single individual analysis. Because if we think about it, like for a pair of gene and paralogs, then normally we're looking at 4 different haplotypes. So if it's a mixture of 10 individuals, then that would be a very large number of haplotypes to look at. And this is not something that is currently designed for, and it could be, yeah, it could be problematic and not that easy to interpret.
Yeah. All right. Does Paraphase work with 5MC data? Is it possible to get the allele of chromosome X inactivated?
So currently for PacBio data, the methylation is like naturally available with the data, but so the reads do encode that kind of information, but for Paraphase we don't use that kind of information. But I imagine that with the chromosome X inactivated, it shouldn't have any impact on Paraphase analysis.
All right. Another question, can Paraphase replace Sanger sequencing completely for CYP21A2, or can we use it only for haplotyping?
So our idea is that with Paraphase we can do a comprehensive analysis for CYP21A2. We can call the copy number, we can identify all the pathogenic variant, any kind of fusion, gene conversion. So we think that it can replace Sanger sequencing completely.
To be tested, I suppose, locally. Can Paraphase support other diploid species? Any plans for this functionality?
Currently, we're just working on the human genome, and we're still working actively to add like segmental duplications that are medically relevant into Paraphase, and we expect that it will take some time before we add all the interesting regions from the human genome into Paraphase. So at this point, I think we won't be able to support other diploid species in the near future, but we might consider it when we kind of finish up on the human genome.
All right. One more question, I believe, and then we're close to the end. What's the maximum insert size for PCR-based target capture? Can we target for above 25 kb? That's a bit on the high end, and we typically see 5 to 10 kb type insert sizes. Can go a bit broader and larger than 10 kb, up to 15, but that's stretching it. Most of the insert sizes are around 5 to 10 kb.
There's one last question about TTN.
Yeah.
The question is, what about TTN gene? Have you explored this technology for it? So Yeah, we are aware of the homology in TTN. So we haven't added this gene yet, but we'll take a closer look at it. My understanding is that it has sequences with similarity, but that homology region is just about 3 kb long. So if this is the homology region that you're talking about, my guess is that because HiFi reads are already 15 to 20 kb long, so HiFi reads are long enough to cover that homology region, as it's just about 3 kb. So we might not need any specialized tool to analyze the TTN gene, but we will definitely take a closer look at it.
Wonderful. Another question, what was the insert size for the RCCX module?
So if this is for Paraphase, Paraphase works on whole genome sequencing data and hybrid capture-based enrichment data. The read length for whole genome sequencing is about 15 to 20 kb long. The read length for hybrid capture-based enrichment data is slightly shorter. It's about the data that I have seen is about 5 to 6 kb long. So that's kind of the read length that we are talking about. And for amplicon-based approaches, I believe Jeff might have more information about it, but I think the insert size for the size for the amplicon, it would be about like 10 to 15 kb, and that would cover the CYP21A2 gene, and also might cover some bit of the flanking regions as well.
The data you've shown, Zhao, for RCCX was whole genome, right?
Yes.
Yeah, just to clarify. Okay, I don't see any new questions coming in, and we are very close to the end of the hour, so I think we can wrap up the Q&A here. So I'd like to thank you, Zhao, for your time today and sharing all your experience and expertise. I'd like to thank all the attendees for joining us today, all the great questions we've received. In closing, 3 little points. First, in the next few days, we'll send you an email with a link to this webinar in case you want to listen to it again. Secondly, we are visiting more than 30 cities across North and South America and Europe in the next several months, as you can see here, to bring you captivating peer-to-peer discussions, talks from leading scientists, and critical updates on the latest of PacBio methods. You can scan the QR code on this slide or visit pacbio.com/events for more information. And finally, immediately following this webinar, you'll receive a very brief questionnaire. Please do take the 2 minutes required to fill out, as it helps us to understand your needs and plans for future webinars. So thanks again for joining us today. Hope you'll join us again for a future webinar. And with that, take care and have a wonderful day.
Thank you.